Okay, we're now in Unit 5 of Blood Bank, last unit of the semester. And in this last unit, some of this information that we're going to talk about kind of goes back to some of the topics or the same concepts as in Unit 1. So in this unit, we're going to be talking about serology testing, hypersensitivity reactions and autoimmune diseases, and transplantation immunology, and then molecular diagnostic techniques. So this very first video is, I imagine, is going to be a little bit of a longer one. We're talking about serology and then the autoimmune diseases in this first video. The next one will cover actually, transplantation immunology is not going to be until the third video, I think. So this first one's just on the serological testing and autoimmune. Okay, so the first assays that were developed to detect either antigen or antibody were based on principles of either precipitation or agglutination. Both of those are considered unlabeled assays because there's not a marker label that's needed to detect the reaction. So there's not like an additional label that indicates the reaction. We see the reaction without that marker label. So precipitation in this methodology of precipitation, this combines soluble antigen with soluble antibody to produce insoluble complexes that are visible. And there's 3 zones of precipitation reactions that can occur, and we've talked about these before. So zone of equivalence, that's the point in that antigen-antibody reaction where an approximately equal number of binding sites are present, so that results in optimal antigen-antibody binding reaction. So that's optimal precipitation. Prozone is when there is an excess of antibody present in that reaction, so it results in a lot of free molecules remaining in the solution. You don't have those cross linkages that form, so that precipitation reaction is undetectable. And then you've got post-zone, which is an excess of antigen in the solution. So all of the available antibody sites are bound, but then you have additional leftover antigen. So again, no cross-linkages are formed, no lattice network formed. So again, precipitation does not occur. So in both prozone and post-zone, this can lead to false negative results. Okay, affinity. We've also, I think in Unit 1 we talked about the concepts of affinity and avidity. So with affinity, this is the initial force of the attraction that exists between a single Fab site on an antibody molecule and a single epitope or determinant site on the corresponding antigen. So the strength of the attraction depends on the specificity of the antibody for that particular antigen. So it goes back to the idea or the concept of the lock and key when we're thinking about antigen and antibody reactions. So a key has to be specific for the door or the lock that it's going to open. So you can kind of think about that as affinity, and then avidity represents the overall strength of the antigen-antibody binding. It's the sum of the attractive forces of all the individual antibody antigen combining sites. So it's a measure of the overall stability of that antigen-antibody complex, or the force that keeps the molecules together after that binding has occurred. So a high avidity can compensate for a low affinity. And then different classes of antibodies are going to differ in their avidity. So IgM is going to have higher avidity than IgG because of those number of potential binding sites. So remember, an IgM antibody is a pentamer, so it has 5 binding sites. So even if think about it this way, even if the fit is not perfect, there's more chances for it to hold on versus an IgG molecule, which only has 2 binding sites. Okay, and then agglutination reaction. So this is visible aggregation of particles such as antigen caused by a combination with the specific antibody. So antibodies that cause these types of reactions are called agglutinins. So this is a 2-step process, and again, we've talked about this process before. So a 2-step process that results in the formation of a stable lattice network. So that first stage is sensitization. That involves the antigen-antibody combination through single antigenic determinants on the particle. So this process of sensitization is rapid, but it's also reversible. So this is affected, the sensitization phase of agglutination is affected by the nature of the antigens on the agglutinating particles. So if epitopes are sparse or they are obscured by other surface molecules, then they're going to have less likelihood of interacting with their antibody. And then red cells and bacterial cells have a slightly negative charge, so they tend to repel one another. So because of that, sometimes it's hard to bring those cells together. And then the class of the antibody is important. IgM can bind more sites than the IgG, and they can bind more easily because it's a larger molecule, so it's able to bridge that gap between the cells easier. So just think of, not only does the IgM molecule have more arms, but the arms are longer, so they have more reach. And then the second stage is the lattice formation. So this involves the development of the cross-links that form those visible aggregates. So this represents the stabilization of the antigen-antibody complexes with the binding of multiple antigenic determinants. So you've got that stable network that forms, which is the visible agglutination. This is insoluble. Okay, and then you can look at this table. It just outlines or identifies the major differences between agglutination and precipitation reactions. One of the big ways to think about the differences in agglutination and precipitation reactions are these bottom 2 characteristics here. So in agglutination, the antigen is going to be in sediment form, whereas in precipitation it's going to be in solution form. And then agglutination reactions are commonly used to determine blood groups, so because we're seeing the result of that antigen-antibody combination in the form of agglutination, whereas precipitation reactions are used to determine the presence of a certain element in a solution. So that an example of that would be pregnancy testing. And then here's a table just to represent that precipitation curve, and then those the zone of equivalence versus zone of postzone and prozone, where you have either antigen or antibody excess. Okay, so some precipitation testing, automated methods. So we have immunoturbidity, which is a measurement of the turbidity or the cloudiness of a solution as those immune complexes are forming. So you've got a detection device that measures the reduction in light intensity caused by reflection, absorption, or scatter of light, and it records those things in what they call absorbance units. So you can just think about if you've got a solution and then you've got all these particles in that solution that start forming these networks and start cross-linking and start combining together, the more of that happens, the more turbid the sample is going to become. So you've got this device that measures that reduction in light, because as you've got those precipitates that start forming, less light is going to get through. So that's what it's measuring. Then in nephelometry, it measures the light that is scattered at a particular angle from the incident beam as it passes through a suspension. So the scattering occurs when a beam of light passes through a solution, and then the beam of light encounters a molecule in its path, so it's going to bounce off the molecule. So light is measured by an instrument called a nephelometer. The amount of that light scatter is proportional to either the size, shape, and concentration of the molecules in a solution. So this is similar to, I don't remember what unit it was, maybe unit 1, when we talked about the principles of the basics of flow cytometry. Okay, so here's just a kind of a visual example of what's happening in either nephelometry and immunoturbidity. So you've got these particles that are in a solution, you've got your light source that's passing through that solution. So when it comes in contact with the particles, it's measuring that light, either the reflection of the light or the amount of light that's able to pass through the solution. Okay, some different types of precipitation reactions. So passive immunodiffusion techniques involve the precipitation of an antigen-antibody complex that can be detected by manual methods that use some type of support medium, such as a gel, like an agar gel. So agarose, which is derived from seaweed, is used for this purpose a lot of times. So when the antigen-antibody reactions diffuse toward one another in that gel matrix, you're going to have visible lines of precipitation that form. The agarose helps just stabilize that diffusion process, and it allows us to be able to visualize those precipitation reactions. So with radial immunodiffusion, this is a single diffusion technique that involves migration of antigen only. So you've got a reagent antibody that's uniformly mixed into that support gel or that support agar. Then the antigen that's contained in commercial standards, either a commercial standard or the patient sample, is applied to wells that are cut into the gel. Then there's an incubation step. So during that incubation, you've got the antigen that diffuses out from each of those wells and combines with the specific antibody that's in the agar to form rings of precipitation around the wells. So those rings are going to expand in size until that zone of equivalence is reached and a stable lattice network is formed. And then you can measure that area of the ring, so it gets measured, and that correlates to the antigen concentration in a particular well. And I don't know if you remember, we talked about this in micro a little bit, some different techniques that relate back to this testing methodology. Okay, Auchterlonie double diffusion. So in this type of testing, you've got both antigen and antibody that diffuse independently through a semi-solid medium in 2 dimensions, so that they're diffusing both horizontally and vertically. So again, you've got wells that are cut into the gel, and the reactants are added to the wells. Most Auchterlonie plates are going to be set up with a central well that's surrounded by 4 to 6 outer wells that are equidistant from the center well. Then you've got antibodies that are placed in that center well, antigens are placed in the surrounding wells, and then you've got those diffusion reactions that are going to start to radiate from the wells. Again, this one requires an incubation step, and you're going to look for precipitation lines to form where the moving front of antigens meet the antibody, and where that point of equivalence is reached. So the density of the lines reflects the amount of immune complex that is formed in the reaction. Okay, and then the precipitation of the bands allows for the antigens to be compared with one another and the 3 precipitation bands that are possible. So you've got what's referred to as identity, where you've got fusion of the lines at their junction to form an arc, represents serological identity or the presence of a common epitope. Then you've got non-identity, which is a pattern of crossed lines that demonstrates 2 separate reactions and indicates that the compared antigens share no common epitopes. And then you've got what's referred to as partial identity. So this is indicated by the fusion of 2 lines with what they call a spur. So in the case of partial identity, you've got the 2 antigens that share a common epitope, but some antibody molecules are not captured by the simpler antigen, so they're going to continue to travel on through the initial precipitation line to combine with additional epitopes found in the more complex antigen. So because of that, the spur always points to the simpler antigen. And this is what those 3 different reactions would look like. So this is what they're calling the spur in the case of partial identity. And this is the same information that I just described to you. And then here's just kind of a different way of representing that, the idea of the 3 possibilities, or the 3 possible outcomes with the Auchterlonie double diffusion. Okay, electrophoretic techniques. So diffusion can be combined with the process of electrophoresis to speed up or sharpen the results. So electrophoresis separates molecules according to differences in their electric charge when they are placed in an electric field. So you've got a direct current that's forced through the gel, causing either antigen, antibody, or both to migrate across the gel, and as precipitation takes place, you're going to see those precipitation bands that are formed. So in immunofixation electrophoresis, you've got that initial electrophoresis step that's used to separate the proteins in patient's serum in a gel medium. And typically, the patient's serum is going to be applied in 6 lanes of the gel. So after electrophoresis takes place, 5 lanes are overlaid with 1 of each of the following reagent antibodies. So you'll have antibody to gamma, alpha, or mu heavy chains, and then to kappa or lambda light chains. The 6th lane is overlaid with antibody to all serum proteins, so it's just going to serve as a reference lane. And then reactions in each of the 5 test lanes are compared with that reference lane. Immunoprecipitates form only where you've got a specific antigen and antibody combination that has taken place and the complexes have become trapped in the gel. And then the gel gets washed to remove any non-precipitating proteins, and then this whole process can be stained for easier visibility. So this is what, once it's stained, this is what a result might look like. And then here's a table that just kind of outlines some of the techniques that we just talked about. So the principle in the test and different applications that you could use those tests for. Okay, the next types of agglutination reactions. So everything that we just talked about was types of precipitation reactions. Now we're talking about agglutination reactions. So direct agglutination occurs when antigens are found naturally on a particle. So this is called hemagglutination when that agglutination reaction involves red cells, such as what we do in ABO testing. Passive agglutination, also referred to as indirect agglutination, uses particles that are coated with antigens not normally found on their surfaces. So for example, latex agglutination kit tests, which there are a number of them that we use in the lab. So with those types of tests, a latex passive agglutination test for CRP would use the CRP antibody on the surface of a latex bead, and then you mix that with the patient serum and observe for agglutination. So they use these latex beads just kind of as a carrier for whatever it is that they're trying to detect. Reverse passive agglutination. So in this one, you've got antibody rather than antigen that's attached to a carrier particle, so that latex particles coated with antibody are reacted with a patient sample containing the suspected antigen, and then agglutination occurs. So this has been used in kit tests for the detection of infectious agents such as Group A, Group B, Staph aureus, rotavirus, and Cryptococcus neoformans. It's also been used for measurement of therapeutic drugs, hormones, haptoglobin, and CRP. Agglutination inhibition is based on competition between particulate and soluble antigens for limited antibody combining sites. Haptens are complexed or associated with proteins then attached to a carrier particle. Then you've got the patient sample that reacts first with a limited amount of reagent antibody that is specific for that hapten being tested. Then you add indicator particles that contain the same hapten to be tested for in the sample. So if the patient sample has no free hapten, the reagent antibody is able to combine with carrier particles and agglutinate. So agglutination is a negative reaction indicating that the patient did not have sufficient hapten to inhibit that secondary reaction. And then no agglutination is considered a positive reaction in this type of testing. So this indicates that the patient sample contains a sufficient amount of hapten to bind to the reagent antibody and inhibit agglutination. Antigen or antibody can be attached to the particles. You see this type of testing a lot of times used for drugs of abuse. And I think you guys, if you have been in urines already, then you would have done this test as a lab, or you may have done it this semester. But with these types of reactions, you kind of have to be careful when you're interpreting and reporting them out, because the end result of the test seems kind of counterintuitive to what you think. And then hemagglutination inhibition uses the same principle, but with red cells as the particles. So examples of this type of testing are for the detection of viruses like rubella, influenza, and RSV. Okay, labeled immunoassays. So this type of testing allows for much greater sensitivity than unlabeled immunoassays, which are your precipitation and agglutination testing, because substances can be detected in much lower concentrations. In this type of testing, the substance being measured is often referred to as the analyte. So immunoassay labels, you've got the detection molecule included in the test system. Sometimes it's referred to as the tracer molecule. And then you've got some different ways that this can happen. So colorimetric, or the chromogenic substrate, indicates that there's a production of a pigment. It may be in the form of a fluorescent compound, which is referred to as a fluorophore. You may have luminescent molecules, so they produce chemiluminescence, which is emission of light from a chemical reaction, or radioactive isotopes, but those types of reactions are not usually used in testing today. So some general immunoassay formats Okay, so you're dealing with separation of bound and free tracer labels before signal measurement. So this is a requirement in all immunoassay designs. So that can be broken down as either heterogeneous or homogeneous or competitive, non-competitive. So heterogeneous, you've got physical separation, which is done by centrifugation. So the result of that is binding to a solid phase material or through magnetic separation. Homogeneous is methods that do not require that physical separation, but the method by which the detection, signal, and patient analyte concentration can vary a lot depending on the manufacturer of the test. Okay, and so then we're talking about competitive versus non-competitive. So with competitive type assays, you've got all reactants that are mixed together. Then you've got a labeled tracer antigen that competes with an unlabeled patient antigen for a limited number of binding sites for the antibody. So the concentration of the labeled analyte is in excess to make sure that all of those binding sites on the antibody are going to be occupied whether or not the sample antigen is present. So you've got a wash step that removes unbound label, and then the amount of antibody bound label is measured and used to determine the amount of patient antigen that's present. If the patient antigen is present, some binding sites are going to be occupied with the unlabeled analyte, so it's going to decrease the amount of bound label detected. And then the amount of bound label is inversely proportional to the concentration of the labeled antigen, which means that the more labeled antigen that is detected, the less antigen present in the patient sample. So you just think about it as two different teams that are competing for the same goal. So you've got the labeled and the unlabeled. They're both competing for the antibody. So that's competitive. So everybody's kind of thrown into one mix and they're left to fight it out. With non-competitive, you've got a reagent antibody, sometimes also referred to as a capture antibody, is first passively absorbed onto a solid phase material. Excess capture antibody is present to make sure that any patient antigen present is going to be bound. So you've got unknown patient antigen that reacts with or is captured by the solid phase antibody. It's got a wash step to remove unbound antigen, and then a labeled antibody directed against a different epitope on the antigen gets added to the reaction. So the amount of label measured is directly proportional to the amount of patient antigen present. So it's represented by a mathematical linear relationship. So, and then here's kind of the visual representation of competitive versus non-competitive and how those things are reacting. We're going to talk about a few different types of labeled immunoassay tests. So the first one, radioimmunoassay. So this is the original immunoassay based on a competitive principle. It used radioisotopes developed in the late 70s. So you've got radioactive elements that decay into other elements and release energy, and that can be measured. So you've got radio-labeled antigens can be tagged with various radioactive elements. The most popular was iodine-125. You've got detectors that are able to measure very low quantities of radioactivity. It gives the assays extremely high analytical sensitivity, so that's good for detection of certain analytes, such as hormones, that would normally be found in low concentrations. But of course, the disadvantage is the health hazards of working with radioactive substances. And then the fact that there's radioactive waste disposal that has to be considered, and the limited shelf life of some of the elements. So this is not a method that's used as much anymore. Enzyme immunoassays. So enzymes are naturally occurring molecules that catalyze biochemical reactions. Enzyme immunoassays use enzymes as the labels. Enzymes convert reagent substrates to produce chemically modified products that can be detected, such as colored or visible light, UV light, fluorescent light, and luminescence. Advantages of this method is that one molecule of enzyme can generate a lot of molecules of product, so that improves sensitivity, and then you don't have any concerns over disposal. There are no health hazards with this one. Some common enzymes that are used as labels for this type of testing are horseradish peroxidase, alkaline phosphatase, beta-degalactosidase, and glucose 6-phosphate dehydrogenase, G6PD. Competitive enzyme immunoassays. So you've got enzyme-linked immunosorbent assays, or ELISA, where you've got sample antigen that competes with enzyme conjugated antigen for a limited number of binding sites. So again, you've got two different things competing for the same target. So limited number of binding sites on antibody molecules attached to a solid phase, such as plastic tubes or microtiter wells. This involves a wash step to remove unbound antigen. Enzyme activity present in the final reaction catalyzes the conversion of the substrate to a detectable product. And then the product signal is inversely proportional to the concentration of the test substance. So in practice, an ELISA is the same as an EIA, so you might see those terms used interchangeably. Okay, non-competitive enzyme immunoassays, so those ELISAs or EIAs, can also be based on non-competitive design, where they can be used to detect the presence of either an antigen or an antibody in the test sample. So immunoassays that detect antibody are most commonly indirect ELISAs. Those that detect antigen are commonly called capture immunoassays. So in addition to microtiter plates, solid phase supports like plastic reaction well tubes, plastic beads, magnetic particles, and latex particles can all be used. They're most commonly used to detect a patient antibody that's being tested for, and they're called indirect because the enzyme labeled reagent does not precipitate in the initial antigen antibody reaction. So the associated antigen is bound to the solid phase, and patient serum that has the unknown antibody that you're trying to find is added, and then there's an incubation step. So after it's incubated, there's a wash step. An enzyme labeled antiglobulin, which is a secondary antibody, is added, which reacts with any patient antibody that is bound to the solid phase. So if no patient antibody is bound to the solid phase, the second labeled antibody will not be bound and will be removed during a second wash step. This gets followed by an incubation with the enzyme substrate and a stop solution that stops that enzyme activity. Then the amount of color, fluorescence, or luminescence generated by action of that enzyme label on the substrate is measured using a detection device and compared with known amounts of antigen. Signal detection is directly proportional to the amount of antibody in the specimen. So if the color reaction phase is prolonged, though, the result is going to be falsely increased and vice versa. Okay, capture or sandwich immunoassays. You've got antibody bound to a solid phase. Antigens captured in these immunoassays have to have multiple epitopes. Excess antibody attached to the solid phase is allowed to combine with the test sample to capture any antigen present. Then you've got an incubation period. Then there's an enzyme labeled antibody that is added. That second antibody recognizes a different epitope or binding site than the solid phase antibody. And then the final complex that's formed with the sample antigen is formed in between the two reagent antibodies. So that's why it's called a sandwich assay, because it kind of creates a sandwich. Depending on the enzyme used, either a colored, fluorescent, or chemiluminescent reaction product is going to be detectable. The product formation is directly proportional to the amount of antigen present in the test sample. And capture immunoassays are best suited to antigens that have multiple determinant sites. So that would be such as cytokines, proteins, tumor markers, microorganism antigens. There's just a visual on the concept of the sandwich immunoassay. Okay, chemiluminescent immunoassays can be used for detection of antibodies or antigen. This is commonly used for therapeutic drug monitoring to detect steroid hormones, protein hormones, antibodies produced in infectious diseases and autoimmune disorders. So they perform well at very high and very low concentrations, meaning that they have the testing method has a high sensitivity, which is important in detection of tumor markers and autoantibodies. Then fluorescent immunoassays, you've got antibodies or antigens can be labeled with molecules that fluoresce. And then these compounds are called either fluorophores or fluorochromes. So you've got a direct immunofluorescence assay or DFA used to detect antigens on tissue sections that are fixed onto a microscopic slide or in live cell suspensions. The antibody that is conjugated with a fluorescent tag gets added directly to the sample antigen on the slide. Most common fluorescent tags used are fluorescein and rhodamine. You've got an incubate and a wash step. The wash step removes unbound antibody. Then you read it under a fluorescent microscope. The bound fluorescent probe is detected under UV light. So antigens are usually seen as bright apple green or orange yellow objects against a dark background. This method is commonly used for detection of Legionella pneumophilia and Chlamydia trachomatis. Okay, indirect immunofluorescence assays can be used for antigen or antibody detection. In the first step, you've got the patient serum that is incubated on a microscopic slide where you have a known antigen that has been attached. The slide is then washed and an anti-human globulin containing a fluorescent tag is added. So that labeled immunoglobulin combines with the first antibody to form a sandwich, which localizes that fluorescence, makes it easier to see. Multiplex immunoassay is an automated method where you've got polystyrene beads that are used as the solid phase. When they are used for detection of antibodies, the beads are going to be conjugated to different antigens, and then the patient sample is added to the bead mixture, and antibodies in the sample are detected with fluorescent tagged anti-human immunoglobulin. and then the beads containing the immune complexes are identified through flow cytometry. Fluorescence polarization immunoassays was discontinued. This method was discontinued in 2008. It was based on the change in polarization of fluorescent light emitted from a labeled molecule when it's bound to an antibody. The degree of polarized light detected corresponds to the amount of antibody-labeled antigen complex formed in a solution, and the fluorescence polarization detection is inversely proportional to the sample analyte concentration. Okay, then we've got radioimmunoassays. So these are membrane-based tests. They're easy to perform. They give reproducible results. They're designed primarily as point-of-care testing type tests, but they are used in the clinical lab because most of them are going to have fast turnaround times. They don't take up a lot of space. They're easy to use. They're intended as single-use tests, so they are disposable. And they're based on what is called a flow-through design, where you've got antigen or antibody in the patient sample is absorbed onto the membrane that contains the corresponding antibody or antigen. There's a wash step, and then the reagent is added, and the reaction is read by looking for the presence of a colored reaction product. So usually it's just going to be like a line on the little test cartridge. Newer immunochromatography methods combine all of the previously mentioned steps into one step. So you've got an analyte that's applied at one end of the strip and it migrates toward the end of the test where there is an absorbent pad to maintain the constant capillary flow rate. The labeling and detection zones are set between the two ends, so the end where you put the patient sample and then the end that it migrates to. When the sample is added to the application point, which also contains a labeled antigen or antibody conjugated to a colored latex, the sample reconstitutes the conjugate, so it forms a complex that migrates across the membrane. An antigen or antibody immobilized in the detection zone captures the immune complex, forms a colored line for a positive test when the test is non-competitive. So this is the methodology behind, when we do pregnancy tests, strep tests, point of care, troponins. So that's the idea behind those types of tests. Some newer test designs use a competitive format where the absence of a line indicates a positive result. So that's the one I mentioned just earlier in this lecture that's commonly used for drugs of abuse testing. Okay, and then flow cytometry, we've talked about flow cytometry before. This is an automated system where you've got single cells or beads in a fluid suspension get analyzed in terms of their intrinsic light scattering characteristics and extrinsic properties such as surface or cytoplasmic proteins using fluorescent labeled antibodies or probes. So that's referred to as gating. Different populations of cells or particles can be analyzed and identified on the basis of their size, shape, and antigenic properties. One of the biggest advantages of flow cytometry is that because the flow rate of cells within the cytometer is so rapid, thousands of events can be analyzed in seconds. So it allows for the accurate detection of cells that are present in very small numbers. Some more common applications for flow cytometry are in leukemia and lymphoma characterizations, identification of immunodeficiency diseases like AIDS, in detecting human leukocyte antigen antibodies for transplantation. And then the major components, and I just realized I've got a duplicate slide. The major components of a flow cytometer include the fluidics, the laser light source, and the optics and photodetectors. And then you're going to have a data analysis and management system that's performed in the computer. So you've got cells processed into a suspension. They get passed through a laser light one at a time. Each time this happens, the laser is interrupted and the signal is recorded where forward scatter and side scatter are measured. So forward scatter is an indicator of size, side scatter is an indicator of granularity and intracellular complexity. And then those two forward and side scatter characterize the different cell types based on their inherent properties. So those are what are considered your intrinsic parameters. And then extrinsic parameters require addition of a fluorescent probe for detection. So you've got fluorescent labeled antibodies bound to the cell that are detected with the laser. And again, I think we talked about this in Unit 1. And you probably talked about it in hematology also. There is a video, YouTube video in the class shell in Moodle that you can look, but this is the basics of the components of a flow cytometry system. So this is going to be the basic idea behind the hematology analyzers that you use in the lab. So that's the end of this first lecture video for Unit 5. The next one we're going to get into hypersensitivity and autoimmunity.